The fog you're experiencing right now, the inability to focus, the mental exhaustion that no amount of sleep fixes, it's not in your head. It's in your cells. Specifically, it's happening inside the tiny power plants called mitochondria that exist in every single neuron in your brain. And here's what nobody tells you. The damage is already done. Every neuron in your brain contains between 1,000 to 2,000 mitochondria. These microscopic structures produce something called ATP, adenosine triphosphate, the actual currency of cellular energy. Your brain is only 2% of your body weight, but it consumes 20% of your total energy. That energy comes from one place, mitochondria. And when those mitochondria are damaged, when they can't produce ATP efficiently, your neurons don't fire properly. You can't concentrate. You can't remember. You feel like you're thinking through mud. Brain fog. But here's the terrifying part. The most common causes of mitochondrial damage are things you're doing every single day. Things your doctor probably told you were healthy. Things that are hiding in your kitchen right now, slowly destroying your brain's ability to produce energy. Stay with me, because what I'm about to show you isn't about age. It isn't about genetics. It's about a biological process happening in real time inside your neurons. And the moment you understand what's actually happening, everything changes. Let's start with what your mitochondria are supposed to do. Inside every mitochondrion is a process called oxidative phosphorylation. This is where your cells take the food you eat, specifically glucose and fatty acids, and convert it into ATP through a complex series of reactions involving 5 protein complexes embedded in the inner mitochondrial membrane. Complex 1 takes electrons from NADH. Complex 2 takes electrons from FADH2. These electrons move through complex 3 and complex 4, pumping protons across the membrane and creating an electrical gradient. That gradient powers complex 5, ATP synthase, which literally spins like a turbine, converting ADP into ATP. Your brain uses roughly 5.6 milligrams of ATP per 100 grams of brain tissue per minute. That's approximately 1.5 kilograms of ATP per day in an average adult brain. And all of it comes from this process. When it works, you're sharp. Your memory is clear. You process information quickly. Your mood is stable. You have mental endurance. When it doesn't work, everything slows down. The problem is that mitochondria are incredibly vulnerable, more vulnerable than almost any other cellular structure. And the reason has to do with their location and their evolutionary origin. Mitochondria used to be free-living bacteria. Around 2 billion years ago, they were engulfed by our cellular ancestors and became part of eukaryotic cells. They still have their own DNA, mitochondrial DNA, separate from the nuclear DNA in your chromosomes. But that DNA is sitting right next to the electron transport chain, right next to the location where electrons are being passed from complex to complex. And sometimes, those electrons leak. When they do, they react with oxygen and create something called reactive oxygen species, ROS. Superoxide, hydrogen peroxide, hydroxyl radicals. These are not metabolic byproducts you want floating around. They're highly reactive molecules that damage everything they touch: proteins, lipids, DNA. And the first thing they damage is the mitochondrial DNA itself, because of its proximity to the electron transport chain. Mitochondrial DNA doesn't have the protective histone proteins that nuclear DNA has. It doesn't have the same robust repair mechanisms. So when ROS damages it, that damage accumulates. Mutations build up. And damaged mitochondrial DNA leads to dysfunctional electron transport chains, which leak even more electrons, producing even more ROS. It's a vicious cycle. The oxidative damage isn't limited to DNA. It hits the proteins of the electron transport chain directly. Complex 1 becomes inefficient. Complex 3 leaks more electrons. ATP production drops. And when ATP production drops in your neurons, you get brain fog. Studies published in the Journal of Clinical Endocrinology found that individuals with cognitive decline showed significantly reduced mitochondrial function in brain tissue, decreased ATP production, increased oxidative stress markers, damaged mitochondrial membranes. But here's the critical question. What's causing this damage in the first place? Your mitochondria are designed to handle a certain amount of ROS. They have antioxidant systems. Superoxide dismutase converts superoxide into hydrogen peroxide. Glutathione peroxidase neutralizes hydrogen peroxide. Catalase breaks it down into water and oxygen. Under normal conditions, these systems keep oxidative stress in check. But something in the modern environment has pushed oxidative stress beyond what your mitochondria can handle. And the most insidious part is that it's not just one thing. It's a cascade of factors that amplify each other. Let's talk about the first villain, the one hiding in plain sight: linoleic acid. Linoleic acid is an omega-6 polyunsaturated fatty acid. It's the primary fat in industrial seed oils, soybean oil, canola oil, corn oil, sunflower oil, safflower oil. These oils didn't exist in the human diet before the 20th century. Now they're everywhere. Salad dressings, restaurant food, processed snacks, baked goods. Between 1909 and 1999, linoleic acid consumption in the United States increased by 136%. And this isn't a minor dietary change. Linoleic acid incorporates directly into your cellular membranes, including the membranes of your mitochondria. The inner mitochondrial membrane is made up of a specialized phospholipid called cardiolipin. Cardiolipin is essential for the structure and function of the electron transport chain. It holds the protein complexes in the correct configuration. It facilitates proton and electron transfer. And the fatty acid composition of cardiolipin is determined by your diet. When you consume high amounts of linoleic acid, it gets incorporated into cardiolipin. And here's the problem. Linoleic acid is a polyunsaturated fat with multiple double bonds. Those double bonds are chemically unstable. They're highly susceptible to oxidation. When linoleic acid in your mitochondrial membranes gets oxidized, it produces a toxic compound called 4-hydroxynonenal, 4-HNE. 4-HNE is one of the most damaging lipid peroxidation products in biological systems. It forms adducts with proteins, covalently binding to them and altering their structure. It damages the electron transport chain complexes directly. It breaks ATP synthase. It inhibits pyruvate dehydrogenase, the enzyme that converts pyruvate into acetyl-CoA for the TCA cycle. Research published in Free Radical Biology and Medicine demonstrated that 4-HNE exposure in neuronal cultures led to immediate mitochondrial dysfunction. ATP production dropped by 40% within hours. Electron transport chain efficiency plummeted. Neurons couldn't maintain their membrane potential. And the damage compounds. When mitochondria are damaged by 4-HNE, they produce more ROS. That ROS oxidizes more linoleic acid in the membranes, producing more 4-HNE, damaging more proteins, creating more ROS. It's a self-perpetuating cycle of mitochondrial destruction, and it's happening in your brain right now if you're consuming seed oils regularly. Studies on patients with neurodegenerative diseases published in Frontiers in Pharmacology found elevated levels of 4-HNE in brain tissue, specifically in regions associated with memory and cognition, the hippocampus, the prefrontal cortex. These aren't just correlations. Animal studies have shown direct causation. Mice fed diets high in linoleic acid from soybean oil developed greater insulin resistance, fatty liver, and cognitive impairment compared to mice fed saturated fats from coconut oil. The mechanism? Oxidative stress induced mitochondrial dysfunction in the brain. But seed oils aren't the only problem. There's another factor most people overlook: glucose instability. Your brain runs on glucose. Neurons take up glucose and convert it through glycolysis into pyruvate, which enters the mitochondria for oxidative phosphorylation. When glucose delivery is stable, mitochondrial function is stable. But modern diets create glucose chaos. High sugar foods, refined carbohydrates, frequent snacking. These create rapid spikes in blood glucose, followed by insulin surges, followed by crashes. Your blood sugar swings wildly throughout the day. And here's what that does to your neurons. When glucose levels spike, excess glucose enters neurons. That glucose gets metabolized rapidly, flooding the electron transport chain with electrons. The system becomes overwhelmed. Electrons leak. ROS production skyrockets. Research in the journal Frontiers in Endocrinology showed that even 6-hour fluctuations in glucose levels caused decreased mitochondrial activity in neural cell cultures and activated intrinsic apoptotic pathways. Brain cells started dying. Chronic hyperglycemia does something even worse. It causes glucose to react with proteins in a process called glycation, forming advanced glycation end products, AGEs. These AGEs bind to mitochondrial proteins, damaging the electron transport chain complexes and impairing ATP production. Studies published in the Journal of Clinical Investigation found that individuals with poorly controlled blood sugar had significantly higher levels of oxidative damage in brain tissue. Specifically, they found increased levels of 8-oxodeoxyguanosine, a marker of oxidative DNA damage in mitochondrial DNA. But it's not just high glucose. Low glucose is equally damaging. When your blood sugar crashes, neurons experience energy deprivation. They can't produce enough ATP to maintain basic cellular functions. Ion pumps fail. Membrane potentials collapse. Neurons become vulnerable to excitotoxicity. During hypoglycemia, the brain attempts to compensate by increasing glucose uptake. But this creates another problem. The sudden rush of glucose when levels normalize triggers a reperfusion-like injury, flooding already damaged mitochondria with substrate and causing an oxidative burst. The solution isn't just avoiding sugar. It's about glucose stability, preventing the spikes and crashes, maintaining a steady supply of energy to your mitochondria. But even if you fix your diet, there's a third factor that's destroying your mitochondrial function, and it's one that most people don't even realize is a problem: chronic low-level inflammation. Your immune system is supposed to protect you, but when inflammation becomes chronic, when it's constantly activated at a low level, it attacks your own tissues, including your brain. Inflammatory cytokines, molecules like TNF-alpha, IL-6, and IL-1-beta, cross the blood-brain barrier. They activate microglia, the brain's immune cells. Activated microglia release more inflammatory mediators. They produce nitric oxide and superoxide, which combine to form peroxynitrite, an extremely reactive oxidant. Peroxynitrite damages mitochondrial membranes. It inhibits complex 1 and complex 4 of the electron transport chain. It causes mitochondrial DNA strand breaks. Research in Nature demonstrated that chronic neuroinflammation led to progressive mitochondrial dysfunction in neurons. ATP levels dropped. Membrane potential decreased. Neurons couldn't clear damaged mitochondria through mitophagy, the cellular process that removes dysfunctional mitochondria. Instead, damaged mitochondria accumulated. They became sources of even more ROS production. They released mitochondrial DNA into the cytoplasm, which activated inflammasomes, protein complexes that trigger more inflammation. Another vicious cycle. And here's the connection most people miss. The factors I've been describing don't operate in isolation. They amplify each other. Seed oils high in linoleic acid don't just damage mitochondria through oxidation. They also promote inflammation. Linoleic acid is metabolized into arachidonic acid, which is converted into pro-inflammatory eicosanoids. These inflammatory mediators activate immune cells and increase cytokine production. Glucose instability doesn't just damage mitochondria through oxidative stress. It also triggers inflammation. High glucose activates NF-kappa B, a transcription factor that turns on inflammatory genes. It increases production of inflammatory cytokines. And inflammation doesn't just damage mitochondria directly. It also impairs glucose metabolism and increases lipid peroxidation. They're interconnected. Each one makes the others worse. This is why brain fog isn't just about being tired. It's not about needing more sleep or drinking more coffee. It's about a fundamental breakdown in your neuron's ability to produce energy. Let me show you exactly what that looks like inside your brain. Your prefrontal cortex, the part of your brain responsible for executive function, attention, and decision-making, is one of the most metabolically active regions. The neurons there are constantly firing, processing information, maintaining working memory. Every action potential, every synaptic transmission requires ATP. Lots of it. When mitochondrial function is optimal, those neurons have the energy they need. Information flows smoothly. You can focus. You can think clearly. You can switch between tasks without mental fatigue. But when mitochondrial damage reduces ATP production, even by 20 or 30%, everything slows down. Neurons can't maintain their resting membrane potential efficiently. Action potentials become less reliable. Synaptic vesicles don't release neurotransmitters as effectively. Receptors aren't recycled quickly enough. The subjective experience is brain fog, difficulty concentrating, slow processing, mental fatigue. The feeling that your brain just isn't working the way it should. And the worst part is that this damage is cumulative. Every day that your mitochondria are exposed to excess ROS, to linoleic acid peroxidation products, to glucose fluctuations, to inflammatory cytokines, more damage accumulates. Mitochondrial DNA mutations build up. You can't reverse those mutations. They're permanent. Proteins in the electron transport chain become increasingly dysfunctional. Even if you remove the damaging factors, those proteins are already damaged. They need to be replaced through mitochondrial biogenesis, the creation of new mitochondria. But here's the problem. Mitochondrial biogenesis is itself an energy-intensive process. It requires ATP. And if your existing mitochondria are damaged and can't produce enough ATP, you can't make new healthy mitochondria efficiently. You're trapped. This is why brain fog often gets worse over time, why people who struggle with it find that no amount of rest helps, why cognitive function continues to decline even when they're doing everything they think is right. They're treating the symptoms, not the cause. So let's talk about the solution. The first step is eliminating the damage. You have to stop poisoning your mitochondria. Get rid of seed oils completely. Read labels: soybean oil, canola oil, corn oil, sunflower oil, safflower oil, grapeseed oil. These are not health foods. They're industrial products that didn't exist in significant quantities in the human diet until recently. Replace them with stable fats: olive oil, coconut oil, butter, ghee, tallow. These fats are either monounsaturated or saturated. They don't oxidize easily. They don't produce toxic lipid peroxidation products. Research published in the Journal of Lipid Research showed that replacing polyunsaturated fats with saturated and monounsaturated fats reduced markers of oxidative stress and improved mitochondrial function in animal models. The change isn't optional. If you want your mitochondria to recover, you have to remove the source of ongoing oxidative damage. Second, stabilize your glucose. This doesn't mean going low carb or keto unless you want to. It means preventing the wild fluctuations that damage mitochondria. Eat protein and fat with every meal. They slow glucose absorption. They prevent spikes. Avoid refined carbohydrates and added sugars: bread, pasta, pastries, soda, fruit juice. These cause rapid glucose spikes followed by crashes. Time your meals consistently. Don't skip breakfast and then binge at dinner. Your mitochondria need a steady, predictable supply of substrate. Studies in the Journal of Clinical Endocrinology showed that individuals who maintained stable blood glucose had significantly better cognitive function and lower markers of neuroinflammation compared to those with high glucose variability. Third, reduce inflammation. This is more complex because inflammation has multiple sources, but there are key strategies. Eliminate processed foods. They're loaded with inflammatory compounds: advanced glycation end products from high heat processing, trans fats, emulsifiers that disrupt the gut barrier. Increase omega-3 fatty acids, fish, cod liver oil. These produce anti-inflammatory mediators that counteract the effects of omega-6 metabolites. Get adequate sleep, 7 to 9 hours. Sleep is when your brain activates the glymphatic system, a waste clearance mechanism that removes inflammatory debris and damaged proteins from brain tissue. Studies published in Science showed that glymphatic clearance is 60% more active during sleep than during wakefulness. When you don't sleep enough, inflammatory waste accumulates in your brain. It damages mitochondria. It impairs cognition. But eliminating damage is only half the solution. You also need to actively repair your mitochondria and promote the growth of new, healthy ones. This is where things get interesting. Mitochondrial biogenesis, the creation of new mitochondria, is regulated by a protein called PGC1 alpha, peroxisome proliferator activated receptor gamma coactivator 1 alpha. PGC1 alpha is a master regulator. When it's activated, it turns on genes that code for mitochondrial proteins. It increases the expression of antioxidant enzymes. It promotes the synthesis of new mitochondrial membranes and the replication of mitochondrial DNA. More PGC1 alpha activity means more mitochondria, healthier mitochondria, better energy production. So how do you activate PGC1 alpha? Exercise. Specifically, the type of exercise that creates metabolic stress in your muscles and forces your body to adapt by building more mitochondria. High-intensity interval training. Resistance training. Any form of exercise that depletes ATP and creates a temporary energy deficit. When your muscles run low on ATP during exercise, they activate AMPK, AMP-activated protein kinase. AMPK is an energy sensor. When the ratio of AMP to ATP rises, AMPK turns on. And one of the things AMPK does is activate PGC1-alpha. This triggers mitochondrial biogenesis, not just in your muscles, but throughout your body, including your brain. Research published in Cell Metabolism showed that exercise increased PGC1-alpha expression in the hippocampus, the brain region critical for memory formation. Mice that exercised had more mitochondria in their neurons, higher ATP production, and better cognitive performance on memory tasks. But here's the key. The exercise has to be challenging enough to create metabolic stress. A leisurely walk won't do it. You need intensity. You need to push your muscles to the point where they're gasping for energy. That's what triggers the adaptive response. 20 minutes of high-intensity intervals 3 times a week is enough to significantly increase mitochondrial biogenesis. Sprint intervals, hill sprints, kettlebell swings, anything that makes your muscles burn. The second way to activate PGC1-alpha is through cold exposure. When you expose your body to cold, your mitochondria have to work harder to generate heat through a process called non-shivering thermogenesis. This is mediated by uncoupling protein 1, UCP1, in brown adipose tissue. UCP1 allows protons to flow back across the mitochondrial membrane without producing ATP. Instead, the energy is released as heat. This might sound wasteful, but it has a powerful side effect. It activates PGC1-alpha. It increases mitochondrial biogenesis. It improves mitochondrial efficiency. Studies in Nature Medicine showed that regular cold exposure increased mitochondrial density in brown fat, improved glucose metabolism, and enhanced insulin sensitivity. You don't need to take ice baths. Cold showers work. 30 seconds to 2 minutes of cold water at the end of your shower is enough to trigger the response. The third way to support mitochondrial function is through specific nutrients that enhance ATP production and protect against oxidative damage. Coenzyme Q10. CoQ10 is a critical component of the electron transport chain. It shuttles electrons from complex 1 and complex 2 to complex 3. Without adequate CoQ10, electron transfer slows down, ATP production decreases, and electrons leak, producing ROS. Your body makes CoQ10, but production declines with age. By age 40, your mitochondrial CoQ10 levels can be 30% lower than they were at 20. Supplementing with CoQ10, specifically the reduced form called ubiquinol, restores levels. Studies published in the Journal of Clinical Biochemistry and Nutrition showed that CoQ10 supplementation improved mitochondrial function in aging adults, increased ATP production, and reduced markers of oxidative stress. Dosage: 100 to 300 milligrams per day of ubiquinol. Next, NAD+, nicotinamide adenine dinucleotide. NAD+ is a coenzyme involved in hundreds of metabolic reactions. It's essential for the function of the electron transport chain. It's required for sirtuins, proteins that regulate mitochondrial health and longevity. NAD+ levels decline with age. By age 50, your NAD+ levels are roughly half of what they were at 20. Low NAD+ means impaired mitochondrial function, reduced ATP production, decreased ability to repair damaged DNA. You can increase NAD+ by supplementing with precursors. Nicotinamide riboside, nicotinamide mononucleotide, NMN. Research published in Cell demonstrated that NMN supplementation restored NAD+ levels in aged mice, improved mitochondrial function, and enhanced physical endurance. In human studies, NMN supplementation improved insulin sensitivity and increased muscle mitochondrial oxidative capacity. Dosage: 250 to 500 milligrams per day. Third, magnesium. Magnesium is required for ATP synthesis. ATP exists in cells as a complex with magnesium, Mg-ATP. Without adequate magnesium, ATP synthase can't function properly. Magnesium deficiency is extremely common. Roughly 50% of people in Western countries don't meet the recommended daily intake. Low magnesium means impaired ATP production, even if everything else is functioning correctly. Supplementing with bioavailable forms of magnesium, like magnesium glycinate or magnesium threonate, restores levels and improves mitochondrial function. Magnesium threonate specifically crosses the blood-brain barrier more effectively than other forms, making it ideal for supporting neuronal mitochondria. Studies published in Neuron showed that magnesium threonate improved synaptic density and cognitive function in aging rats by enhancing mitochondrial function in neurons. Dosage: 300 to 500 milligrams of elemental magnesium per day. Fourth, creatine. Creatine isn't just for muscle. Your brain uses creatine to maintain ATP levels during high energy demand. Creatine phosphate acts as a rapid ATP buffer. When neurons fire intensely and ATP gets depleted, creatine phosphate donates a phosphate group to ADP, regenerating ATP instantly. This prevents energy crashes during intense cognitive tasks. Research published in Psychopharmacology showed that creatine supplementation improved memory and reduced mental fatigue in healthy adults. The effect was particularly strong in tasks requiring sustained attention and processing speed. Your brain synthesizes some creatine, but not enough to saturate stores. Supplementation increases brain creatine levels and enhances cognitive performance. Dosage: 5 grams per day. Fifth, pyrroloquinoline quinone, PQQ. PQQ is a cofactor involved in mitochondrial biogenesis. It activates PGC1-alpha independently of exercise or cold exposure. It promotes the growth of new mitochondria and protects existing ones from oxidative damage. Studies in the Journal of Nutritional Biochemistry showed that PQQ supplementation increased mitochondrial content in cells and improved energy metabolism. In human trials, PQQ improved cognitive function in middle-aged and elderly adults, particularly in measures of attention and working memory. Dosage: 10 to 20 milligrams per day. But even with all these interventions, there's one more critical factor: time. Mitochondrial damage accumulates over years. Reversing it doesn't happen overnight. When you eliminate seed oils, it takes months for linoleic acid levels in your cell membranes to decrease. Your body has to gradually replace those membranes with fats from your new diet. When you stabilize glucose, it takes weeks for glycated proteins to turn over and be replaced with undamaged versions. When you activate mitochondrial biogenesis through exercise and supplements, it takes time for new mitochondria to be synthesized, for old damaged ones to be cleared through mitophagy, for the overall population of mitochondria in your cells to shift toward healthier, more efficient ones. You won't feel better tomorrow. You might not feel better next week. But if you're consistent, if you eliminate the damage and actively support repair, the changes compound. After 4 weeks, you might notice small improvements: slightly better focus, less afternoon brain fog. After 8 weeks, the changes become more obvious. Mental clarity improves, fatigue decreases. After 6 months, your cognitive function can be dramatically different: sharper memory, faster processing, sustained mental energy throughout the day. This isn't theoretical. This is what happens when you fix the underlying biology. Your brain fog isn't a mystery. It's not just stress. It's not in your head. It's mitochondrial dysfunction. And now you know exactly what's causing it and exactly how to fix it. The question is whether you're willing to do what it takes. Because the truth is, most people won't. They'll read this, they'll understand it, and then they'll go back to eating seed oils and spiking their blood sugar and wondering why nothing changes. Don't be most people. Your mitochondria are waiting for you to stop poisoning them. The moment you do, the moment you give them what they need to recover, everything changes. Your focus returns, your memory sharpens, the fog lifts, and you realize that the brain you thought you'd lost was there the whole time. It just needed fuel. Now that you understand the basic framework, we need to go deeper, because mitochondrial damage doesn't just cause brain fog through simple ATP depletion. There are multiple interconnected pathways that amplify the dysfunction, and understanding these mechanisms is critical if you want to fully reverse the damage. Let's start with something called reductive stress. Most people have heard of oxidative stress, too many reactive oxygen species. But there's another form of metabolic stress that's equally damaging and far less understood: reductive stress. Your mitochondria rely on a delicate balance between oxidized and reduced states of electron carriers. NAD+ is the oxidized form. NADH is the reduced form. When the ratio of NADH to NAD+ becomes too high, the electron transport chain becomes overloaded with electrons. Electrons back up. They can't be processed quickly enough. And when that happens, they leak from the complexes and react with oxygen, forming superoxide. This is reductive stress. Too many electrons, not enough capacity to process them. And it's caused by the exact factors we've already discussed: high linoleic acid diets, glucose instability, chronic inflammation. Here's how it works. When you consume high amounts of fat, particularly oxidized linoleic acid, your mitochondria shift toward burning fatty acids instead of glucose. Fatty acid oxidation produces enormous amounts of NADH and FADH2, the reduced electron carriers that feed into the electron transport chain. Under normal conditions, this isn't a problem. The electron transport chain can handle it. But when the electron transport chain is already damaged by 4-HNE from linoleic acid peroxidation, its capacity decreases. Complexes 1 and 3 become less efficient. They can't process electrons as quickly, so you have more electrons coming in from fatty acid oxidation and less capacity to process them. Severe reductive stress. Massive ROS production. Research published in Free Radical Biology and Medicine showed that high linoleic acid diets induced reductive stress in liver mitochondria, causing oxidative damage to proteins and DNA, despite the liver not being in a state of classical oxidative stress. The same mechanism operates in brain mitochondria, and it creates another vicious cycle. Reductive stress damages the electron transport chain further, reducing its capacity even more, making the reductive stress worse. The second hidden mechanism involves calcium dysregulation. Mitochondria don't just produce ATP, they also regulate intracellular calcium levels. Neurons use calcium for signaling. Every action potential involves calcium influx. Every neurotransmitter release is triggered by calcium. But too much calcium is toxic. Mitochondria act as calcium buffers. They take up excess calcium from the cytoplasm, preventing it from reaching toxic levels. This process requires a functional mitochondrial membrane potential. The electrical gradient across the inner membrane drives calcium uptake through a channel called the mitochondrial calcium uniporter. When mitochondria are damaged, when their membrane potential decreases, they can't buffer calcium effectively anymore. Calcium builds up in the cytoplasm. It activates enzymes called calpains, which degrade cellular proteins. It triggers excitotoxicity, where neurons become overexcited and die. Studies in neuron demonstrated that mitochondrial dysfunction in neurons led to impaired calcium buffering, increased cytoplasmic calcium levels, and activation of apoptotic pathways. The neurons didn't die immediately. They became dysfunctional first. They couldn't fire properly. They couldn't maintain synaptic connections. Brain fog. And calcium dysregulation amplifies oxidative stress. High intracellular calcium increases the activity of enzymes that produce ROS. It activates NADPH oxidase. It stimulates nitric oxide synthase. More ROS, more mitochondrial damage, less calcium buffering capacity. Another vicious cycle. The third mechanism involves something called mitochondrial dynamics. Mitochondria aren't static. They're constantly fusing and dividing in a process called mitochondrial dynamics. Fusion allows mitochondria to share contents, mixing healthy components with damaged ones to maintain function. Fission allows the cell to segregate severely damaged mitochondria for removal through mitophagy. This balance is critical for maintaining a healthy mitochondrial population. But when oxidative stress is high, when inflammation is chronic, mitochondrial dynamics become disrupted. Fusion decreases. Fission increases excessively. Mitochondria fragment into small, dysfunctional pieces that can't produce ATP efficiently. Research published in Cell Metabolism showed that oxidative stress induced excessive mitochondrial fission in neurons, leading to fragmented mitochondrial networks and decreased ATP production. The neurons couldn't maintain energy homeostasis. They became vulnerable to stress. Cognitive function declined, and fragmented mitochondria produce more ROS. They have less capacity for oxidative phosphorylation. They're more likely to release cytochrome C and trigger apoptosis. Another amplifying cycle. The fourth mechanism is perhaps the most insidious. It involves the gut-brain axis. Your gut microbiome influences mitochondrial function in your brain, and the connection is direct. When you consume high amounts of linoleic acid from seed oils, it doesn't just affect your own cells, it affects your gut bacteria. Linoleic acid alters the composition of the gut microbiome, decreasing beneficial bacteria like Akkermansia and Lactobacillus, and increasing potentially harmful species like Proteobacteria. This dysbiosis impairs the intestinal barrier. The tight junctions between intestinal cells become leaky. Bacterial endotoxin, lipopolysaccharide, LPS, leaks into the bloodstream. LPS is highly inflammatory. It activates the immune system. It triggers the release of pro-inflammatory cytokines, TNF-alpha, IL-6, IL-1 beta. These cytokines circulate through the bloodstream. They cross the blood-brain barrier. They activate microglia in the brain. Activated microglia release their own inflammatory mediators. They produce ROS. They damage neurons and their mitochondria. Studies published in Frontiers in Nutrition showed that high linoleic acid diets induced gut dysbiosis, increased intestinal permeability, elevated systemic inflammation, and caused neuroinflammation and mitochondrial dysfunction in the brain. And it gets worse. Some gut bacteria produce metabolites from linoleic acid that are directly neurotoxic. 10-hydroxy-cis-12-octadecanoic acid, HYA. HYA crosses the blood-brain barrier and impairs mitochondrial function in neurons. It disrupts glucose metabolism. It increases oxidative stress. The result? Cognitive impairment, brain fog. So fixing your mitochondria isn't just about diet and supplements. It's also about restoring your gut health. Probiotics, fermented foods, prebiotics that feed beneficial bacteria. These interventions can reduce gut dysbiosis, decrease systemic inflammation, and indirectly protect brain mitochondria. Research in psychopharmacology showed that probiotic supplementation reduced inflammatory markers and improved cognitive function in adults with subjective cognitive decline. The fifth hidden mechanism involves sleep and the glymphatic system. We touched on this earlier, but it's worth diving deeper because most people don't realize just how critical sleep is for mitochondrial health. During deep sleep, your brain activates the glymphatic system. This is a waste clearance pathway that uses cerebrospinal fluid to flush out metabolic debris, damaged proteins, and inflammatory molecules from brain tissue. The glymphatic system is 60% more active during sleep than during wakefulness, and it's essential for removing the oxidative waste products that accumulate during the day. When you don't sleep enough, when your sleep quality is poor, glymphatic clearance decreases, waste builds up, damaged proteins accumulate, inflammation persists, and this waste damages mitochondria. Beta-amyloid oligomers, proteins associated with Alzheimer's disease, directly impair mitochondrial function. They insert into mitochondrial membranes. They disrupt the electron transport chain. They cause calcium dysregulation. Studies in Alzheimer's and dementia showed that poor sleep quality was associated with higher levels of beta-amyloid in the brain and worse cognitive performance. The mechanism is straightforward. Insufficient glymphatic clearance allows amyloid to accumulate. Amyloid damages mitochondria. Damaged mitochondria can't produce enough ATP. Cognitive function declines. But sleep does more than just activate the glymphatic system. It also allows mitochondria to undergo repair processes. During sleep, autophagy increases. This is the cellular cleanup process that removes damaged organelles, including dysfunctional mitochondria. Mitophagy, the specific form of autophagy that targets mitochondria, is enhanced during sleep. Old, damaged mitochondria are tagged with ubiquitin. They're engulfed by autophagosomes. They're degraded in lysosomes. This clears the way for new, healthy mitochondria to be synthesized. When you chronically undersleep, mitophagy is impaired. Damaged mitochondria accumulate. They produce ROS. They can't generate ATP efficiently. Research published in Science showed that sleep deprivation decreased autophagy in neurons, led to accumulation of damaged mitochondria, and caused cognitive deficits in mice. The mice couldn't learn new tasks. They had impaired memory consolidation. They showed signs of neuroinflammation. All of this reversed when normal sleep was restored. So if you're doing everything right with diet and exercise and supplements, but you're still sleeping 5 or 6 hours a night, your mitochondria can't fully recover. You need 7 to 9 hours, consistently. Now let's talk about one more hidden factor that most people completely ignore. Electromagnetic fields, EMFs. This is controversial, and I'm not going to claim that EMFs are the primary cause of brain fog, but there is emerging evidence that chronic exposure to certain frequencies may affect mitochondrial function. Mitochondria contain iron in the form of iron-sulfur clusters in the electron transport chain complexes. These iron-sulfur clusters are essential for electron transfer. They're also potentially sensitive to electromagnetic fields. Research published in the International Journal of Radiation Biology showed that exposure to radiofrequency electromagnetic fields altered mitochondrial membrane potential in certain cell types and increased ROS production. The effects were subtle, not massive, but when you're dealing with mitochondria that are already damaged by linoleic acid, glucose fluctuations, and inflammation, even small additional stressors can tip the balance. The practical implication? Reduce unnecessary exposure. Don't sleep with your phone next to your head. Use wired headphones instead of Bluetooth when possible. Take breaks from screens. Again, this isn't the primary factor, but it's worth being aware of. The bigger point is that mitochondrial health is multifactorial. There isn't one single cause of dysfunction. There are multiple overlapping mechanisms, and they all interact. Linoleic acid oxidation produces 4-HNE, which damages proteins and DNA, causing reductive stress and calcium dysregulation. Glucose instability creates oxidative bursts and glycates proteins, impairing electron transport. Inflammation from gut dysbiosis activates microglia and releases cytokines, which damage mitochondrial membranes and disrupt dynamics. Poor sleep impairs glymphatic clearance and mitophagy, allowing damaged mitochondria and toxic proteins to accumulate. Each factor makes the others worse. This is why isolated interventions often don't work. Taking CoQ10 while still eating seed oils won't fix the problem. Exercising while sleeping 5 hours a night won't fully restore function. You need a comprehensive approach. Eliminate seed oils, stabilize glucose, reduce inflammation, prioritize sleep, support mitochondrial biogenesis through exercise and cold exposure, supplement with key nutrients. All of it, together. When you do that, when you address every component of the system, the results are transformative. Because you're not just treating brain fog, you're fixing the underlying biological machinery that powers your entire brain. And once that machinery is working again, everything else follows. Your memory improves, your focus sharpens, your mood stabilizes, mental fatigue disappears. You feel like yourself again. But it requires commitment, it requires consistency, and it requires understanding that this isn't a quick fix. This is a months-long process of systematically repairing cellular damage. Most people aren't willing to do that. They want a pill. They want instant results. But mitochondrial dysfunction doesn't develop overnight, and it doesn't resolve overnight. The damage accumulated over years. The repair takes months. That's the reality. The question is whether you're willing to accept that reality and do what it takes. Because the alternative is living with brain fog for the rest of your life, watching your cognitive function slowly decline, accepting that this is just how things are now. You don't have to accept that. Your mitochondria can recover. Your neurons can regain their energy. Your brain can function the way it's supposed to. But only if you give it what it needs. Stop the damage. Support the repair. Be patient. The fog will lift.